Empirical papers means they are based on certain realistic data which you compile through charts, graphs, tables, whatever it is, which you created through systematic experimental methods or whatever it is. Okay. So it is not like writing one novel out of your imagination or anything. It will have some 8 to 30 pages with selected references called bibliography. So it is defined as any research where conclusions of a study, where conclusion of a study is strictly drawn from concrete evidence, and therefore it should be verifiable. If suppose today I publish a paper, tomorrow again if you want to redo the same experiment, and you should get the same results what I got, not like I am getting one result and you will be getting that, and all not permissible, particularly in scientific. What is this? Anybody can identify? Wow. Yeah, my sister. It looks not that attractive here, black and white. But if you see even today it is very beautiful. So how much effort would have gone behind this building? Okay, so much, right? It would have started from the foundation. So what is the foundation for your scientific paper? What is the foundation? Huh? Didn't hear. Objectives. Good hypothesis, right? You should have very good hypothesis. You can prove or disprove. Many times students will think whatever their objective or hypothesis, they have to prove it. No, even disproving it also a good paper. So you can prove it or you can disprove it. That is research paper. Okay. So this is one stanza in Bible actually. See, it says, it is the glory of God to conceal a thing. It is the glory of God to conceal a thing. And it's the honor of kings to search it out. So, who and all are in research? Let it be MSc, PhD, or your faculty, or any full-time researcher like me, all are kings and queens. Right? We are trying to find out what God concealed. Such a high position is given for researchers. Okay? So we should not meddle it. Right? We should not meddle it. We should not cook the data. Okay? Just find out what exactly comes. That is the beauty of research. So what you should do to to have a good hypothesis, you should think differently. But many of my students or whoever it is, I usually see, they will be very disturbed, very annoyed if they get results something different than previously reported literature. They want a statement, my results are coinciding. What is that sentence? Huh? On par. If they won't get that, they will be restless. No, they won't be even confident enough to say, yes, I got this. What is wrong with that? You did with all your honesty. You got whatever you are getting, A plus B, C, whatever it is. So you have to be courage enough to present that. Why you are looking for some XYZ who got different results? So think differently, be confident. Okay, this paper just I want to give you an explanation. Last time also I showed you in chemical ecology, but the context is different here. Actually, I won't work on sericulture things. I never work. I work on horticultural pests. But when I was talking about horticultural pests and oviposition stimulants in one conference, one sericulture scientist asked me, Madam, you are identifying ovipositional stimulants for all pest insects, which are of no use. And if you can identify oviposition stimulants for beneficial insects like silk moths, it will be very good because silk moths die with a good amount of eggs in the abdomen before they release. So if you can stimulate those female moths to release even 1%, that is big contribution to the industry. I never thought in that direction actually. So after I said, okay, maybe it doesn't fall into my mandate. I never thought about it. So when I go back, I thought, okay, let me check. As just out of my own interest, I was started digging out what exactly this silk moth, what he meant. So this paper I found, very good paper. And high-end labs in the world like Max Planck and some Japanese laboratories worked on it. And they said the Bombyx mori, right? Bombay, what is Bombyx mori? Mulberry silk moth. What is the wild progenitor of Bombyx mori? Huh? Mandarina, correct. Bombyx mandarina. So you can see here that brown one, small brown one, that is Bombyx mandarina. It is free-flying insect which lives outdoors. Okay. The Bombyx mori, which we domesticated for thousands of years, forgot to fly, right? It can't live outside. It has to live in the room only. Okay. So what these people did, they conducted one experiment and they exposed some odors. Okay, environment odors. I checked what type of odors they are. And when they exposed Mandarina responded more less vigorously. So what they said, the olfaction ability of Bombyx mori compromised or reduced, whatever it is. So then I thought it didn't sound convincing for me actually. Suppose I like idli or sambar or like you guys. If we give same idli sambar to an American, okay, whether he will relish it? No, right? And can you say his taste buds are compromised? No. So how can they say Bombyx mori olfaction is compromised? So then we thought, what are the odors Bombyx mori is familiar with? What are the odors? Because all the time it is inside the room only. What are the odors they are exposed during rearing stages and all? Hardly mulberry leaves, right? Mulberry leaves were fed. So only that odor may be familiar from larval stage to adult stage. That's it. So I thought, okay, the female, particularly the female insect, while laying eggs, it look what and all parameters it will look. What and all parameters? What and all parameters it will consider to lay egg in particular because the progeny should be safe, right? So, see, your parents would may take how much care they will take about you, right? Huh? They will say whether your hostel is comfortable or your food is okay. Everything they will check, right? In case of Bombyx mori, the Lepidopteran moth, once it lays eggs, the time the progeny or egg emerges, the mother is gone. Mother is not there. Progeny has to survive on its own. So how much care it will take? The place where it is going to lay egg, abundant food supply should be there for the larva because larva can't fly and go. Right? If food is not there, whether it can fly and go? No. So it should be natural enemy free space. So many things. So then I thought, okay, if you see Bombyx mori, egg laying process in the industry, how Bombyx mori eggs were laid? How? One brown sheet will be given, right, for a mated moths. And they cover that too with again one cup, a plastic cup or something, and it will lay eggs within that. Okay, there is no cues, general cues, which a mother moth will look. So we thought, okay, let us give Bombyx mori, the mulberry odors. Okay, and we did GCEAD. Okay, this and all peaks which it responded. And when we took those chemicals, we did a detailed biosis. You can see here total number of eggs laid, number of eggs retained, and hatched eggs. We took unhatched eggs also mainly because Lepidopteran has the ability, trait like to lay unfertilized eggs. So we shouldn't count unfertilized eggs and we should not say more eggs were laid. So all care we took and we found it is responding and it is releasing more eggs. Okay. And it helped us to develop some formulation. It is called Arca Extra. Now we are asking the silk board people to just spray the brown sheet, which gives mulberry odor, and now they are evaluating. So what led to the whole process of one technology? That one person who asked us, can you develop ovipositional stimulants? I think he really, what I should say, sensitized us differently. His thought process was different. than us. So just interact with your peers and discuss the subject. It will give you lot of ideas. So it gave us 3 more projects. And DBT asked us to identify ovipositional stimulants for Muga, Eri silk moth, and Tassa silk moth. Now currently we are working on all these 3. So think differently. Okay. So, and you need not have all the time high-end data. Definitely high-end data will fetch you good rated publications in high-end journals, but not all the time. Because this paper you see, obstacles in IPM adoption in developing countries. Can you imagine where this public paper was published? Can you imagine? Published in PNAS. What is PNAS? Next to Science and Nature. Huh? Proceedings of National Academy of Sciences. If you see that, I kept that. If you see that impact factor, I put there, NOS impact factor. How much? 18 point something, right? Next, Science and Nature are 20. Highest. This is second journal in the top NOS rating. And you see that topic they published. They don't have any high-end molecular work or any high-end instrumentation data. Just they prepared a questionnaire. Why IPM is not being practiced in developing countries? One good questionnaire they made and they distributed across some countries, maybe 100, 125 countries they distributed, and they collected the responses and they analyzed all those responses and they published a paper. Is it sound very, very simple, right? And they could publish in PNAS. Really, all the time I will draw my inspiration from this paper. You need not have all the time high end. Good idea, good data collection, and good analysis. How you present your data. That is more important than type of experiment what you are going to conduct. You see that materials and methods you read out. What it says, a survey was conducted across so many countries and they made one questionnaire. What are the factors affecting IPM adoption? So many things, variables they identified and they asked the person who are answering. Simple. So shouldn't be bothered about all the time. You see, this is again another paper. A field-based quantitative analysis of sublethal, this one, again published in PNAS from my Geeta, from our own student. She went to NCBS and after her PhD, she started working as postdoc in NCBS. Such a beautiful paper. Not much is involved. What they did, just they identified less polluted, more polluted, medium polluted areas within Bangalore. Okay, they sampled honeybees and they collected data on all type of aspects, all type, molecular, everything, structural and all. You see this, video won't run, but that are run separately. Yeah. You see this paper, they dissected out hot. Okay. How many of you noticed hot in insects? How many of you? You know hot in insects? How is hot here that sensitivity of video? They dissected the honeybees hot in high polluted, medium polluted and high areas and they recorded the heartbeat. And they are showing in low polluted area how heartbeat is, in high polluted areas how it is. So publishing PNAS is not big thing. Are you feeling or not? Okay. See, you can publish simple papers like IPM adoption to even much more. You see, highly polluted, how heartbeat is there. This is the video they submitted. All the time I think. So it is not that complicated to publish in PNAS. This is another paper, which our own paper. Actually, what we did, we were trying to generate the transcriptomic data of methyl eugenol exposed fruit flies. So what we are doing, you know, in dipterans, the antenna is very small, right? What type of antenna? Arista, very small. So thousands of antennas we needed to get the transcriptomic data. So finally, my RA is not that, what I should say, huh? Patience. She doesn't have that much patience. So she said, I will chop the head. I said, okay. Because at some point we need to like compromise and say, okay, we will chop the head and we'll generate the transcriptome data. When she was chopping and leaving the headless bodies back, she noticed that those headless females were roaming around very nicely without any much disturbance, and they are laying eggs also. So she came to me, see how without even head also these females are moving around, minimum from a few hours to 7 days. They are live. They are live. They are happily moving around, cleaning the wings, cleaning the legs, everything. And even they are responding to oviposition stimulants. This and all data we generated. They are responding to oviposition stimulant. We prepared one graph, though they are maximum time in the stationary phase like purple, but they are showing all other activities. So this gave us this paper because her small observation, why females without head are moving, is so that gave this paper. So you should be very, very alert while conducting your own experiment. Maybe it may not fall into your routine experiment, but something else. But don't miss the minute details while you are conducting the experiment. This is my data. My PhD work. My PhD work. Okay. I worked in for my PhD in ICRISAT. I did my PhD then APAU, Andhra Pradesh Agriculture University, and I was allotted to ICRISAT to carry out work. So I was doing with ICRISAT host plant resistance, and I was attached to a breeder and entomologist. ICRISAT is really lovely place to work. Okay. And my experiment was line into tester analysis. So I crossed 12 parents, female parents, with 12 male parents, and 144 hybrids it gave me, along with 24 parents. And all these parents have rainy season specific lines, post rainy season specific lines, and also land races. Okay. All these 176 entries, 3 replications, and 2 seasons. In each season, 2 locations. How many entries? And I have to take observations on early seedling vigor, glossiness, leaf traits, egg count, dead hearts, blah, blah, blah. And but they used to give me labor, huge number of labor. But morning itself, I have to go crop work area. I should say this many labor I need for today observations. So huge fighting will be there between permanent employees. There will be technical staff will be there, right? Their routine experiments will be running. I am there like piggybacking on them, student who came from elsewhere, and I am taking all their labor. They will shout at me. One day I started crying in the crop work area only because one research associate, what are you thinking? You are taking all labor. I started crying there only, and somebody saw me and went to told my guide, Jayanti is crying in the crop work area. So immediately I got message, come to chamber. I went. He didn't ask anything. He said, you are here not only to learn research, you are here to learn HR management. Why you are crying? You have to learn what is the skills involved to manage the human resources. This is such a wonderful guide, mentor I had at ICRISAT. Okay. So 2 seasons, 2 locations. You know, in ICRISAT, same place, you have black soils, you have red soils. So in both soils, I have to conduct in each season. It was hectic. It was hectic, I tell you. It was hectic. From morning around 6, I leave my hostel. My hostel is quite far from the ICRISAT. Catch some bus, go and board the ICRISAT bus, reach there by 8 o'clock it starts. And by the time I come back, it will be evening 7. Okay. I never used to see sunrise or sunset in the hostel. Okay. On the roads or in the field only. And but my friend also used to work with me. And what I used to see when crop is not there, I have to take trichome data. I used to store the samples, leaf bits in the lactic acid. And when crop is not there in the field, I used to take all my trichome data in the microscope. She used to tell me, Jayanti, your eyes will go. Okay. These I got only after 40, not at that time. Okay. Your eyes will go. Just put one approximate number. Why sitting with the microscope counting each trichome? Put some approximate number. Get rid of this. So then I thought, so somehow I was not willing to do that. I took the data. And another beauty is, I don't know what is which genotype. Okay. My guide will give the all 176 genotypes a code number. I don't know whether they are susceptible. I don't know whether they are resistant, because they have different groups actually and hybrids. So when I finished my data, I have to just give data back to him, enter in the computer and give back to him. They will decode back what is what, and they will analyze. One day their statistical division analyzed the data. They called me, and my guide was so impressed. Jayanti, such a nice data you got. I was thinking all these API students will come and time pass here. Okay. And but your data shows that trichomes are expression is season specific. If they are rainy season resistance, the trichomes are expressing only in rainy season. If they are post rainy season specific, it's vice versa. I was so impressed. But my attempts also didn't go waste. That thesis gave me best thesis award in the university, and also Indian Science Congress Young Scientist Award. So don't hesitate to give 100%. Okay? You will be rewarded at any one point of time with your efforts. Don't get influenced or hesitate. And another thing, yeah, be curious. Whatever you see, be curious what you are observing, what you are noticing. Okay? Why I am telling this? This is the experiment which I ran in UK, Rothamsted, for fruit fly isolation and identification of cues. While I was doing this, all this bioassays, to be frank, I tell you, my life as postdoc in other countries, let it be UK, let it be USA, worse than student. I have to wash my glassware. I have to clean my cages. Everything. Actually, researcher life in India is really better when compared to other countries. Not much support. You have to do everything. And but that gives you a type of discipline actually. So when I was doing all these things, I noticed some of the chemicals when I am exposing the fruit flies, they started laying eggs in F and F tubes itself. I don't know why they are releasing eggs in the F and F tubes when I am exposing them to certain chemicals. That observation, but I couldn't do there in UK. When I came back, I conducted experiments in India, and it gave me several papers. Just that observation, several papers it came out. So be curious to your experiment, and understand what is happening there. Okay? And don't forget to upgrade yourself all the time. All the time. People who don't upgrade themselves will become obsolete, particularly in today's era. So when I joined ARS and IIHR, we were not exposed to biotechnology, DNA, RNA, all these things. We never even had practicals also. So first time I was hearing in after joining, I thought unless I understand the techniques, I may not able to have confidence to interact even with my peers. So I applied for to upgrade myself. I applied for Boy Scout fellowship, and I went to US, and just to learn DNA isolation. Okay, I did molecular characterization of fruit flies there. What you will do in molecular characterization? Yes, DNA isolation, run with some MCO1 primers. Your job is done. For that, you need a Boy Scout fellowship to go to US, sir. But in those days, they gave because the biotechnology was coming up. Okay, and it was really tough decision for me in those days. Just I applied for Boy Scout fellowship, and parallelly, my mother was searching a groom for me. And by the time I got married, I got my child, and my Boy Scout results came. You are selected for Boy Scout. And I have my kid, not even 2 months old. I wrote to DST like this. I'm having small kid. Please postpone my fellowship to next year. I want to go next year. They said, what they would have said? The fund is dedicated for this financial year. If you want to use before 31st, you use it. Otherwise, you apply fresh next year. I don't know what to do. I was just quietly watching. My husband was broad-minded. He didn't say no at least. And he said, but he didn't show me the alternate way how I can go, leaving 2 months old kid. So my mother came to my rescue. She said, I will take care of your kid. You go. It was very, very tough decision. Even till now, my daughter rags me. She's now in second year medicine, but she rags me. Who, you heartless mother, she says. When she want to fight with me, she takes that. And she calls my mother as Amma. Okay, I am Jayanti Amma. My mother is Amma for her. Okay, but some tough decisions you have to take, but keep upgrading yourself from time to time. Otherwise, you won't be in the race. I tell you. 2004-5, I went and learned. And this project, I got Lal Bahadur Shastri Young Scientist Award because of that trend. Next, once I came back and I submitted one project proposal, which earned me this one. Because the molecular exposure which I had there gave me confidence to propose a project which has a molecular basis. So I studied the molecular basis of foraging behavior in cryptolemurs. So never hesitate to upgrade yourself. Never ever. Okay. So now you know the molecular biology, right? Now there is no ecology. It is like now genes in ecology, ecology is in genes. So that much integral molecular work it has become such an integral part of ecology now. So you know this guy? Painter. Okay. Who is he? Huh? Host plant resistant. I am fortunate enough when I was in Kansas, I could walk into his lab and I could see his handwritten papers, which they just preserved there. Painter, he was in Kansas State University. Before him, there was no host plant resistance, right? He is father of host plant resistance. So many things he did. See, he developed the world first course on host plant resistance and the first book on host plant resistance again, and so many things. See, here he says, those characters. Which characters? That traits which give or impart host plant resistance to plants. Those characters that enable a plant to avoid, tolerate, or recover from attacks of insects under conditions that would cause greater injury to other plants of same species. That is host plant resistant, he said. But you see how much criticism he got. This all his observations or discussions were printed in proceedings of Association of Applied Biologists, like our minutes of meeting. Can you say what they said? Orange painter of Kansas to be convinced that it is only necessary to walk through the infested crop and pick out the resistant plants. And all this is nonsense. That's what breeders are telling. Whatever he is proposing, host plant resistance, like preference, non-preference, anti-biases, all these are bullshit. You just walk through the plants and pick the resistant ones. That much criticism he got. But he never went back. He had courage to say what he wanted to say. So, yes, facilities do matter, but it is not all that, right? I showed you in the beginning itself. Even IPM paper with a questionnaire can be published. But when facilities are there, please make use of it. Definitely, it will add color to your day work. He did without any facility. Who is he? Vitandate, who identified first sex pheromone in silk moths, Bombicol. He didn't had any experiments or any GCMS, GCAD. He didn't had anything, right? Just his observation. Why male moths are aggregating on spots where female moths were left previous day. That only gave him idea. There is some chemical which female is secreting and that is attractive to male counterparts. That gave the first sex pheromone in the silk moth, bombicol. So you need to have observing skills, not instruments or anything else. The first thing you need is observation and interest in your work, what you are doing. You know, anybody know about these things? This generation? What are they? Oh my God. How come? Ah, is it? Wonderful, yeah. That's why I always admire TNU. My students never know what is that. I said, that's my own handwriting. I sat in library, wrote bundles. Why? I don't know why I am collecting also many times. Because my guide says I will go and write. Okay. But it helps. Because, yeah, if you don't read, you will get extinct. Right? So spend time with literature. It will give lots of ideas, lots of ideas. Okay. And writing and publication of empirical papers, particularly in high indexed journal. Okay. So what is the reason to publish? Why to publish? Time pass, huh? Huh? Why to publish? You conduct study and not report the finding. Is it not crime? Is it not crime? Just I came to know you people are getting very handsome fellowship. Right? All are getting handsome fellowship. Huh? That is taxpayers' money, right? When we are using taxpayers' money and doing our own fancy things, whatever study results we get, we have to publish it. Like, otherwise, it is crime. Okay? And another thing again, it comes whether the results are worth reporting. So that depends on you only. What type of work or what type of experiment you are planning. It needs to be strong enough and interesting enough for the audience to read and the editor to accept. So the results and the study should be worth reporting. So what is difference between scientific writing and science writing? That's just this one. Same thing. Both are explaining the Newton law only. Okay. How the first, here the first panel, the audience is scientists. Okay. So all that jargons will come. Okay. You just confuse your peers with your jargons. Same thing if apple is falling on the head, that is also Newton law. Right. So the difference between scientific writing and science writing. The second panel, it is general audience. You have to tell them like story which they can understand. But you can confuse your peers in scientific writing. That you can enjoy. So, scientific writing is easy? Or writing Agatha Christie novel is easy? Which is easy? Huh? Novel. Somebody is telling novel. Huh? Writing scientific paper is much easier than writing an Agatha Christie novel, right? See how much she should have imagination to write a novel. Such a successful writer, Agatha Christie. How much imagination. And for your scientific writing, just you will conduct one experiment, collect data, put it systematically. That is scientific writing. Okay. So what your scientific writing will conduct? All your IMRs give introduction to your topic so that it will be interesting enough for audience to read through. Some boring introductions I will get from my students, I will share with you. Okay? Methodologies, results, discussion, done. That is scientific paper. So, regardless of the genre, all scientific writing will have same goal. What it is? To present the data with a level of details that you generated to evaluate validity of your idea. Right? So you will be presenting that to your scientific community. But many times it will be boring to sit with a paper. Right? Suddenly, it will be easy to wash all those utensils and get my desk ready than sitting with a paper. Yes or no? How many yes? How many yes? but it is not all that tough. And you can enjoy your writing also. And the first thing you have to do for your paper, identify the right journal. Identifying the wrong journal also, just you will be wasting your time. First, identify the right journal which fits to your work or the general tips which I am giving. Begin with an end in mind. You just understand what you are going to tell in the conclusion. According to your conclusion, your introduction should come, right? And accordingly, the protocols, ideas, outcomes, figures, graphics, all these are coloring things. I will just share my student work here. He worked on okra shoot and food product. Okay, PhD student. His thesis title is Deciphering the Chemical Cues Underlying Trophic Interactions Involving Okra and its Host plant, okra and insect and parasitoids. So, one rule is there, maybe here also. Before thesis submission, they should have one paper, right? It is there in GKVK, US, Bangalore also. So, what they will do, what I should say, that Uppugi Chapani type thing. Some data will be there, right? That data they will keep for that journal. Okay, all heavy data which they want to publish, that they won't give. So, according to their university, they have to publish in Mysore Journal of Agriculture Sciences. One paper should go there. So, this objective, he said, Madam, I will publish this in Mysore Journal of Agriculture Sciences. I said, okay. This is just screening all these genotypes along with some wild species. He did with Irias vitella, choice essay, no choice essay. And along with that, he collected some data on morphological traits and also biochemical. And he want to write paper. So, what I said, begin with the end, right? So, first thing you have to do is give unique title and well informative abstract. I enjoy giving title for my papers actually. I sit with that like as if I am naming my baby. It is so many permutations, combinations I will be doing. Even I will discuss with my kids and I will be whether this is good or this is good. All that stuff I will do. So give very, very attractive title and unique title. And I will show you my student. He gave the first title. Okay. Number of eggs laid by gravid moths and host plant phenology models for population prediction of okra shoot and fruit borer. I was confused. That thesis never talks about population prediction. How he entered into population prediction, I don't know. Okay. Finally, when that paper was accepted, the title was different. What you feel? Whether it is better than the previous one or so? Better, right? And he confused me so much. And why the confusion happened? Because he has to do some correlation and regression in that paper. Because he is taking morphological traits, he is taking biochemical traits. So I asked him, you check which are the traits influencing the egg laying behavior of moths. So he has to do some correlation analysis. So what I did, I shared this paper with him just as a model. You check in this, I did correlation and regression. Maybe it will help you. Gone case. This I used for population prediction. So he ventured into population prediction in okra shoot and fruit borer. There happened the mistake. Okay. And another thing, many times the editors particularly, if you are lucky enough, they will sit with you. If your paper is good, but not in good shape, they will sit with you and they will write the paper along with you. That happened with me in this paper, which I showed in crop protection. That person, see, I said the authors are grateful to the director IAH and the authors duly acknowledge constructive comments of anonymous reviewers and editors. Editors, every time it comes back with the revision, one big para, he scolds me. English is not good, that is not good. And he suggests again. And again I will revise and again I will give back. Oh my God. So if you are lucky enough, the editor will sit through. Otherwise, rejected. Right? And so this is the paper which I shared with him. And my objective here was to predict whether the fruits, guava fruits, can support the fruit fly population. Oh my God, that boy, you see, first introduction, this came to me. The genus Abelmoschus includes a well-known tropical vegetables. Means you can do curry, rasam with vegetable. How entomology student can write all this? You are worried about insect and that thing. Why all this stuff? Okay, I have to sit with him. You see, this is the first introduction which he gave me. I sat with him. I asked him to revise. Then that came. Which is better? The first one says bendi is good for rasam, sambar, the nutrition is this. Why an entomologist needs all those things, right? He is talking about insect plant interactions. He should talk only stick to that. If it is Tamil Nadu is the biggest producer of bendi, how does it matter? How does it matter? Same thing. The objectives, everything one by one. It was really, I said, pathetic to go through, sit through. Okay. And the ability to describe ideas, some people are really blessed enough to do this. I want to again introduce my student. Okay, he's he published this paper. I didn't even correct a single word. Okay, very recent paper in Journal of Chemical Ecology. He worked on it. Now he is a scientist at an ICR Directorate of Medicinal and Aromatic Plant. He first he came to me and he said very looks very flamboyant, easygoing type. And he when he started writing thesis, he started giving me chapters. Okay, beautiful chapters. I first time I am very pessimistic type. Okay, first thing I came to my mind whether he cut and paste. Okay, I asked on his face only. Divija, did you cut and paste? No, madam, he said. Then I didn't believe. I checked with all softwares. Nowhere it is matching. Such a beautiful expression. I never expected that type of paper writing from him. Why I am telling, when he started writing thesis, I asked him, yesterday night I was sharing with Kannan. Okay, because many students won't tell me whether they are in race for their gold medals. Many times they will be running for gold medals. And thesis submission is one criteria which decides on time submission. So many won't tell me, last minute they will come, madam, I am in gold medal race. I have to submit thesis by this. By the time hardly 1 week could have been left. And it will be nightmare for me to finish the thing. So I casually asked him, Divija, are you in any gold medal race? He said, no, madam, who will go for gold medal? Huh? I thought in my heart, what is this boy? Such easy going. How he can take, I don't want gold medal. But that, see, each person is different. You can't judge each person by their grades and by their whatever it is. Each person is unique. They have their own way of expressions and things to do. So Divija is one among them, though he didn't have gold medal. Okay, first time, first attempt ARS he got. So he will do what he want. If he don't want, he won't do. That is the beauty. And you see, this is again one more student. He's senior to Divija. Okay. This boy came to me as MSc student. And usually GKVK, what happened? No, they will identify some students and they will send. Okay, Subhash, now he is scientist in potato. First time when he attached it to me, some trips work he was doing. And some 6 months I didn't see this guy. And suddenly one day came with all his face down like this. What happened, Subhash? I asked. Madam, so many problems, personal problems, all financial problems. Okay, what to do? Madam, I want to shift from GKVK to some Mandya, some Mysore, near Mysore College of Agriculture, because it is tough for me to support a hostel, that, this and all. How you can say no, right? It is very sensitive issue. I said, okay. So, madam, might have changed the guide also. I said, okay, go ahead. Then comes, ma'am, please give me a letter to PG Dean. I can't guide Subhash. Ah, I said, it sounds little fishy, right? See, you have financial problems, you have everything, and you are shifting the college. Why I have to give? I can't guide this student. I said, Subhash, you write to PG Dean because of these reasons I am shifting, and my guide also willing to forego me. That I will sign. Then he started crying. No, madam, all are telling, don't go with that, madam, it will take years. Okay, and better you come out of that lab. Otherwise, you will be in the lab only. This is the first paper which what he published with his MSc in arthropod interactions. After that, somehow for PhD also he came to me only. Okay. And during his PhD, he published this paper. And not this, the one which really I love, this paper as scientist. He is working in potato. Very limited data he collected from my lab actually. Out of affection as guide, he would have given me authorship. But very little support he took from my lab. All his field data only. He published in Pest Management Science. Very recent. I am really proud of this boy. But you imagine how much intelligent he was. Okay? I would have almost gone into his basket. This is another paper I want to share. Volatile chemical signals underlying the host plant preferences. Entomologia experimentalis. She is Vanita. Okay? She's such a wonderful student. It is really, you love to work that girl with that girl. I was thinking she finished MSc and she wrote PhD. She got PhD seat also. PhD seats nowadays in very great demand, right? And after getting seat, she dropped. She said, Madam, no, I'm not going to study PhD. So what you are going to do, Vanita? I asked her. She said she went back to her place somewhere here. She started natural farming. Now she is a farmer. Can you imagine? She wrote such a wonderful scientific paper. And yesterday, I was in flight coming to Coimbatore. I got a call from Vanita. Ma'am, I am sitting in your lab. Where are you? I said, I am not there in office today. Ma'am, I got selected as a best farmer from my district. They brought me to IIHR for exposure visit. Where she worked as her MSc student. I was so happy. You see, each person is different. It is doesn't mean you have to write paper, become a scientist or faculty. No, she chose her path. Okay, but be unique in your way, and you understand. See, as a student, your research you will do so much, but you should have consign what will go from this in your paper. You need to separate them as paper wise. What will go, what won't go, you should able to tell the audience. Otherwise, the editor will get confused and even audience also. The same thing what I said earlier. So try to develop multiple drafts. It will improve the quality of your paper. Try to have as many as drafts possible. So rather instead of getting rejected at editor level, better we revise multiple times. Okay. And when your guide asks you to revise and revise, don't get irritated. That is for your own good. He or she is telling. Okay. And use figures and graphics. I really envy this generation because we used to even, at least I used to have some small, what is that software? Word Star or something we used to tell. Word Star, right? Something. Oh, very complicated thing. You need to understand coding and all. You finally one graph will come. And my seniors are they used to even do with the graph sheets, I think. But what not you have today for making your graphs and this thing, you can prepare beautiful graphs. What and all softwares you have. Get acquainted yourself with all the softwares which are available. Really, it will make your results more beautiful, more appealing. Try to use all this. Okay? And this facility is also now there, I think. Right? Don't fall for it. Don't fall for it. Even during my days only, again, night I was discussing. My senior, he was quite good in writing. He used to, he was plant breeding student, PhD, irrespective of subject. If you pay him some 20,000, you have to give literature, review of literature and results in tabular form. He will give your thesis. People who are really lazy enough, no, they can save money from the fellowship. They will give 20,000 and get the thesis done. But no maza in that. Okay, don't fall for that. And now it is instead of such senior chat GPT is there. I myself was very curious. Really, can I write paper using chat GPT? I went and asked chat GPT, give introduction about Bactrocara dorsalis. Really, it gave such a beautiful introduction. When I went to original and checked the similarity, you check zero. But you can you make use of the facilities, but may give work to your own brains to make unique. Okay. So avoid plagiarism. This is really, really for me to go through the paper which whoever writes, either my colleague or my student, if they cut and paste, it will be very hectic for me to correct. It is like vomiting only, to be frank. See, be original. This is the paper. Just my son, when he was 7 years old, I was just want to give him some work because he shouldn't disturb me. And I asked him, write on honeybees, some paragraph you write. That was with his handwriting. I just made for you to make more legible. You see the mistakes and all. He was just 6 or 7 years. You remember that boy with fish in hand? That boy, he was the author. Okay. He was the author for this. You see that honeybee spelling. Honeybee doesn't even gives us honey, but even food to eat and makes the mother nature beautiful. But the main thing, M-A-N-E, main. Okay. But main thing that it gives us is life. So much he wrote. But I am very confident it is 100% plagiarism free. Even if I check, nowhere even one is the person now. Okay, now he is grown up. He will kick me if he comes to know I put his photo. So you need to write all these things like conception, contribution you need to acknowledge. In any high-end journals, if you are submitting your paper, you need to spell out all these things. Who designed the study? Who contributed like what? Who wrote the draft? Who corrected it? Every detail they will ask. You need to be ready for that. Okay? See how we will write the author contributions. It is very interesting to write. And data availability statements, some journals will ask, funding they will ask, supplementary materials, whatever huge data sets which you cannot give in the main paper, just attach it as a supplementary material. That is good enough. Okay? This is the highest. Responding to reviewer. So how much criticism and but I many times I appreciate reviewers. Every revision, the paper quality goes up. Every revision. And the fun is replying to their queries. It will be very, very enjoyable and very challenging also many times. But if you convince the reviewer with your statements, they will accept. And it will happen multiple times. And this is the paper which one of our PhD student, this figure completely came because of editor or reviewer. Because they said this was not part of the paper. And they suggested you should have one hypothetical diagram how virus spreads in the field. After their suggestion, we started sitting with that and we made it. Is it not enriching the paper? Right? Many times they will help. Their comment, we should not get worried. So, what is stopping you? Right? Just enjoy your paper writing. It is like nothing like that. If you can start, the moment your paper is accepted. The other day, one student who accompanied me to in Madurai, sorry, he went and he was giving Annadana contribution. I didn't understand. Suddenly he just, dropped me in one place and he was running for giving Annadana. I was asking the other person where he is going. Ma'am, his paper is accepted. He is giving Annadana contribution. But so lovely, right? Very nice. So all these emotions, feelings you will go through when you submit your paper, but major things are these things, at least for me. Major things. Once my paper is published in black and white, I won't even see at it even once because I'm sure several mistakes will be there, which I will find out after publication. That really becomes a stress for me. So I won't read my paper once it is in black and white. Whatever I missed. That's it. No. So be truthful for your data. Whatever data is there, don't try to meddle with it. Whatever it is, you try to justify and present beautifully. That's all. And we got one experiment in fruit sucking math. Very beautiful experiment. We could work in the lab, but it drastically failed in the field. Last time I highlighted this. But no, don't be ready for rejection whenever you submit a paper. Okay, because it happens quite naturally with high end journals. The rejection is quite natural unless otherwise you make your paper in such a way. Okay, it is really art. And definitely have plan B immediately. Don't get stressed out immediately. If not, many students I will tell, you keep this journal. If it is not there, just go for current science and finish the job. Don't keep the data and the paper with you. Saved lakhs of lives with his discovery, received 6 Nobel nominations but never got one. Okay, Upendranath, who I discovered urea Stibamin for Kala Ajar. Okay, what they say, it is not my statement, what literature says. However, it was suspected that his ethnicity may have been the prominent reason for him being overlooked for Nobel Prize. Okay, this happens even in publications also. Many times. Many times it is very I don't know, we cannot accept it, but racism do exist, particularly in high-end international journals where editorial board members are there. But just face it. Just face it. Okay. And another major problem is retraction. What is retraction? Huh? Why they take back your paper? Somebody would have been falsified, right? Or cooked the data. Now this has become rampant in publication field. And you can see the retraction rate across the global countries. That is the way. Even India exists there. Though it is very rampant in China and North America, still it exists. This is a country-wise. I love to show this slide all the time. Okay. Enjoy. The paper writing is both art and science. Last time also I showed you this. Okay. Write paper, sit calm, quiet, and as if you are writing a poem or story, and with your cup of coffee or whatever you like, and enjoy your data presentation. That is paper writing. Or it is not like my guide is telling something rude, throw on your guide's face and run away. No, you can enjoy the paper writing. Okay. So, writing paper, whether it is innate skill or learned skill. Huh? Both. Both. It can be innate, it can be learned skill also. You need not be Michael Jackson all the time to dance, right? You can learn dance. You need not be like Sudha Murthy. You can write your paper also, whatever you have in your mind. Okay. So take the plunge. Okay.